Good morning, all of you. We were discussing the cells involved in the immune system and we discussed different types of the granulocytes and agranulocytes which are involved in mounting an immunity. So we discussed neutrophil, basophil, eosinophil in last lecture and the details of each type of the WBC type. So we discussed neutrophil, eosinophil and basophil are called as granulocytes which comprises specific percentage in the blood, and whenever their percentage is either increase or decrease, that reflect the blood defects and that also reports the blood disorder. So the percentage and counting of each type of the cell is important in the same way. So we discuss significance of each type of the cell and they releases certain types of the enzymes as well as mediators and which are responsible for inflammation as well as in mounting the allergic reaction as well as they are helpful in increasing the immunity against certain parasitic infection also. So we discussed the agranulocyte type, that is the monocyte, and we know that the monocytes are leucocyte, which are the largest type. These are agranulocyte and having the kidney-shaped nucleus. The monocyte percentage, if we find that is the it comprises 2 to 8% of total WBC count, and high levels of monocyte indicate the presence of chronic infection or it may indicate autoimmunity or blood disorder or cancer. In case of autoimmunity, we discussed that there is a presence of autoantibodies against the self antigen. So in that case also the monocyte number gone increasing and that also indicates sometime the possibility of cancer. Regarding the structure of monocyte, we studied that these are amoeboid in shape with unilobar nuclei. Last time we have discussed the structure of monocyte. Still I am going to repeat it again. The monocyte is agranulocyte. Agranulocyte means what? Granules are not present there. And when we observe the structure of monocyte is amoeboid like amoeba and having the unilobe nucleus or one lobe nucleus is there. And they get originated in bone marrow and their production is also governed by cytokines and colony stimulating factor. So whenever the number of monocyte increases, that indicate chronic infection, viral infection and tuberculosis. So percentage of monocyte we discussed 2 to 8%, but whenever the percentage or number gone increasing, that indicate the possibility of tuberculosis or tubercular infection or it indicate certain type of the virus infection or it indicate the chronic infection or cancer also. Then what is the function of monocyte? What are the different functions of monocyte? Monocytes involve in phagocytosis using opsonizing proteins. So phagocytosis means they are phagocytic cells are scavenger cells and these scavenger cells they are engulfing the pathogen and they uses opsonizing proteins. Sometime whenever there is a infection by certain types of the bacteria that also results in dead cell formation. So what is the function of monocyte? Monocyte remove dead cells from the site of infection. The first function is that phagocytosis, that is engulfment of pathogen by pseudopodia and by using certain opsonizing proteins. See here, the opsonizing proteins are those which are enhancing phagocytosis, which stimulate the process of phagocytosis. So, monocyte, with the help of opsonizing proteins, they are involved in phagocytosis process. Similarly, the second function of monocyte is that they are responsible in removing the dead cells from sites of infection. The dead cell removal is also important because whenever there is infection and especially wound, the healing of the wound is also important. So removal of dead cell from that particular site is done by monocyte. Secondly, they produces tumoricidal mediators. Tumoricidal mediators means the mediator or the substance which can avoid development of tumor or which can destroy tumor development or tumor cells. That is called as tumoricidal. Tumoricidal means lethal or destroying the tumor formation or destroying the tumor cells. Monocyte also produces certain substances. These are called as mediators. And these mediators are tumoricidal mediators. Means they are responsible to avoid the tumor development. And it means that it is initiating the anti-tumor immunity. Whenever there is a tumor development or cancer development, whatever immunity is seen, that is also due to participation of monocytic cells, because the monocyte develop or produce certain mediators which are tumoricidal mediator, and these mediators initiate anti-tumor immunity. Whatever immunity is seen against the tumor development, that immunity is due to monocyte. And it is why you can say that it is one of the function of monocyte. And it also helps to remove the cancer cells. This is very important and significant function of monocyte. Whenever there is a development of cancer or tumor, the cell should be removed. And otherwise, if the cells are not removed, then the cancer may be spread in very short period of time. That means neighboring cell also get develops into tumor cell. So if the cancer or tumor development prevent at that particular site, then it will help in inhibiting the cancer or tumor spreading. So it is necessary to remove the cancer cell immediately. So whenever these cancer cells are removed, then it will be helping in development of immunity against the cancer, as well as it will help in inhibiting the tumor development. So this is the function carried out by monocyte. These cells help to repair damaged tissue by stopping the inflammation. They are going to repair damaged tissue. Monocyte repair damaged tissue. Whatever tissues are damaged or sometime or due to some reason they are not working properly, then that can be repaired. So who are going to repair? Monocytes. The monocyte helps to repair the damaged tissue by stopping the inflammation. Inflammation means swelling. Tumor necrosis factor TNF produced by this cell help in apoptosis. Apoptosis means programmed cell death, automatic cell death. So TNF or tumor necrosis factor also help in apoptosis. That is one of the function of the monocyte. So what we have discussed here, basophil, neutrophil, eosinophil, monocyte. And these cells, they are comprising the myeloid cell type of the immunity. Immunity or immune system we can say. The immune system comprises myeloid cell type and the lymphoid cell type. So myeloid cell type we have discussed three different cells, eosinophil, basophil and neutrophil, which are called as granulocyte. And in agranulocyte we discuss monocyte, its structure and the function of monocyte. Now we are studying the next type of the cell, that is the macrophages. So these are important type of the cells of the immune system, because they are sometimes they are called as wandering histocytes. Wandering histocytes means these cells are continuously wandering or circulating throughout the body. So whenever there is a entry of specific antigen, it is the major function of the macrophages to receive or to take that particular or to carry that particular antigen towards the antigen presenting or processing cell. When the antigen is carried by macrophages and when it is processed by the antigen processing cell along with the other type of the MHC class 1 and class 2 molecules, then only there is a mounting or there is a production of the antibody. So it is not one step process that as soon as antigen enters in our body, there is immediate production of the antibody. It is not one step process. It will require some time period and it is a multi-step process. So when the antigen comes in contact with the vertebrate body, it is necessary that the antigen should be properly conducted towards the proper antigen processing cell, and then after that, after proper recognition of the antigen by that particular cell, then there is a production of the antibody. The production will be starting later on. So this is the macrophages, means it is the wandering histocyte and it plays very significant role in mounting the immunity. because these macrophages are wandering here and there, and they are capturing or they are carrying the antigen towards the particular type of the antigen processing cell later on. So these are involved in phagocytosis and destruction of bacteria. Macrophages, last time we have discussed, monocytes are also involved in phagocytosis. Similarly, the macrophages are also involved in destruction of bacteria and in the process of phagocytosis. Bacterial infection, whenever occurs, the macrophages are the major cells which are responsible for destruction or removal of the bacteria, and thus the phagocytosis process can be enhanced and bacteria can be removed off. So the body get protected from such type of the infections. So these are antigen presenting cells and initiate inflammation. Antigen presenting cell means what? They carry the antigen. When the antigen comes in contact with the vertebrate body, the antigen is presented by the macrophages. So macrophages are called as antigen presenting cells, and they also initiate inflammation. So they are going to enhance the process of inflammation. When the inflammation is there, whenever the capillaries are swelled, then there is an inflammation. So macrophages are also initiating inflammation process. Actually, there are different types of macrophages. They are categorized as alveolar macrophages, Cooper cells, or splenic macrophages. So alveolar macrophages are there, Cooper cells are there, and splenic macrophages are there. Alveolar cells, Cooper cells, and splenic macrophages. These are the three major types of the macrophages. So what is the significance of all this? And what is the function of these particular type of the cells? Alveolar macrophages, they reside in the alveoli and they play important role in non-specific host defense mechanism. Then Cooper cells, they are important because they are removing the protein complex and whatever cell debris is there. So that cell debris is also removed by the Cooper cell. Actually, Cooper cells are living liver lining, and they are phagocytic cells that first encountered the gut derived bacteria and have been described as highly effective filtering system. Alveolar cells or alveolar macrophages, Cooper cells, and splenic macrophages. These are the three major types of the macrophages. Out of these, the alveolar macrophages reside within the alveoli and they play significant role in non-specific host defense mechanism. So what alveolar macrophages are doing? They are playing important role in non-specific host defense. Non-specific host defense, we know very well, whenever the specific host is not there. But against such type of the agent, whenever the defense mechanism involved, it is the non-specific host defense. So last year we have discussed different types of the barriers which are comes under the non-specific host defense. Specific host defense, when we say whenever specific antibodies are produced against the specific antigen, then we can say that specific defense. Whenever general mechanism is there, general barriers are there, then we say non-specific. But these barriers also having role in preventing antigen and preventing bacteria, and our body get protected from infection of such type of the agents. So alveolar macrophages means what? They are residing, or there's that means what is the location of these macrophages? The location is the in the alveoli, and these alveolar macrophages, they are playing significant role in non-specific defense mechanism. Secondly, Cooper cells, as I said, the Cooper cells, they are residing in the liver lining, means in the liver. Liver is covered by particular lining is there. So in liver lining, Cooper cells are present, and these are also phagocytes that first encounter the gut derived bacteria. Whatever bacteria are there from the gut, that bacteria is first encountered by the Cooper cells, and they have been described as a highly effective filtering system. The Cooper cells is playing as a, or it is playing a role as a filter. So whatever bacteria or unwanted agents are there, that can be filtered by Cooper cells. So Cooper cells remove certain protein complex from the blood, as well as whatever cell debris is there, that is also removed from blood flow. This is the major function of Cooper cell. Then the actually when we see three types of the macrophages, what is the role, combined role of this all types of the macrophages? The first role is the phagocytosis, because it is the process, as we know, it is the process by which the cell utilizes plasma membrane to engulf a large particle, and phagocytic cell or macrophages ingest the pathogen by secreting enzyme. We know that whenever the phagocytic cell with the help of photopodia capture the antigen or engulf the antigen, simultaneously they are producing certain enzymes like lysozyme, and with the help of which the antigen will get engulfed, destroyed, and it is converting into phagolysosome. That part we know very well. So this is the process of phagocytosis. This is the major function of macrophages. So macrophages, the first function is to ingest the pathogen by producing enzymes. And they are also involved in activation of cellular and humoral immunity. They are involved, they initiate the activation of cellular, means T cells whenever they are predominantly involved. The immunity is called as CMI, cell mediated immunity. So in the activation of both type of immunity, cellular as well as humoral, the macrophages are involved. Humoral immunity means antibody mediated immunity, AMI. That is the production of antibodies, and this immunity is enhanced by B cell. Cellular immunity is enhanced by T cell. So macrophages, they are functioning in activation of both type of the immunity, cellular as well as humoral type of the immunity. So this is the function. So in the nutshell, we can say that the functions of macrophages, one is the phagocytosis, second one is the activation of both type of immunity, cellular as well as humoral type of immunity. Alveolar macrophages play important role in non-specific host defense mechanism. Kupffer cells, which are present in the lining of the liver, these are helping to remove the protein complex, and whatever cell debris is there from the blood flow, that can be also removed off. So this is regarding the functions of all types of the macrophages. Thank you.